Welcome. Our speaker today is Malcolm Maden, who is a professor in the Department of Biology. He came to Florida in 2008 from a position at King's College, London, where he taught anatomy and conducted research on the role of retinoic acid in neurological and pulmonary diseases. Prior to that, he worked at the National Institute for Medical Research in London on regeneration of vertebrate limbs. And his PhD was in genetics from the University of Birmingham on the subject of axolotl limb regeneration, which some of his students have continued on with in recent years. And since being at Florida, he and his students have discovered this extraordinary regenerative abilities of a mammal, and namely the spiny mouse, which can regenerate hair, skin, muscle in a completely scar-free manner. So it's a very interesting new line of research, but that's for another day. Today, he will be talking to us about axolotls. Thanks.
Thank you, Jane. Good. Well, thanks for the invitation to tell you about this amazing creature, which actually I've been working on since my PhD 50 years ago. And we've just finished another project on these axolotls. And so, of course, they're amazing. And the reason is that they can regenerate everything: limbs, brain, spinal cord, peripheral nerve, skin, lower jaw, teeth, retina, optic nerve, absolutely everything. That's why we all use them. And of course, people criticize us because they're a long way away from mammals. And are we ever going to learn anything by studying these organisms? But they're still fantastically interesting. I'll tell you in a minute why it's a godlike organism. But the unique feature of this model organism, you know about model organisms from this course, is that it's the oldest laboratory continuous procreating laboratory organism model in the world, because it's been going for 150 years, and I'll tell you about that as we go through. So the unusual thing about axolotls, seen in the previous picture, and that was a white one. This is a wild type, which is gray-green, the color of mud at the bottom of lakes. And here's another side view. They're members of the salamander newt complex, and there are plenty of newts outside here in our ponds. But the unusual thing about them is that they don't metamorphose. So frogs and newts are laid as tadpoles in ponds. They normally metamorphose, lose their gills, and then if you're a frog, you'll lose your tail as well, and you'll jump about outside and reproduce as an adult. But the unusual thing about these guys is they become sexually mature in the larval stage. And that's another reason why they're famous, is because they're a famous evolutionary novelty. So this is a great mechanism of how you can evolve new species. You become sexually mature in the larval stage. So these keep their gills, they never leave the water, and they have become axolotls, and they become axolotls. And probably the reason maybe for that is because they evolved in a lake, one lake in Mexico, which did not dry up in the summer. So people think that amphibians and maybe ordinary salamanders evolved because lakes would dry up or ponds would dry up. And then if they'd metamorphosed, they could walk out on land and solve the problem. But these didn't do that, and they stayed in this one large lake in Mexico and became sexually mature in the adult form. So here's a newt, which this does not turn into, but becomes sexually mature. However, you can make it metamorphose and make a completely new species if you want before your eyes. It's pretty amazing. And so here's a ordinary wild type axolotl in a tank. If you feed it thyroid powder, which you can buy from chemical suppliers, or I don't know how else presumably you from a health food shop, I don't know. But then you'll transform, because the thyroid gland is the thing that makes amphibians metamorphose, and the axolotls have a deficient thyroid gland. So if you give them thyroxine, which you bought in their food, you can turn them into these guys, which look much more like salamanders. So this does not happen naturally. Actually, it rarely happens naturally, but you can make it happen. And you create this new species in front of your eyes. It's pretty impressive. Okay, so it's the oldest self-sustaining laboratory animal in the world. It's been going for 150 years now, and it's got a really interesting history. They come from one lake outside Mexico City called Lake Xochimilco, the only place in the world where they're found. So they evolved here outside Mexico City. And they're very good at eating tacos. So how did it come about? So back in the days of Aztecs, outside Mexico City, there was this huge lake. Mexico City is close by, but this was absolutely massive. And it was settled by the people that the Spanish called the Aztecs. And they built a powerful empire centered here on Lake Xochimilco. And they built the city surrounded by water, not in the middle of the lake, but on the edge of the lake. And they had these walkways going off to the land. But they built this beautiful lake-based city in Lake Xochimilco. It's an island built in the middle of a lake. And they built floating gardens, and it was apparently a very beautiful, beautiful place. And it turns out this is where axolotls come from. And the lake was absolutely jam-packed full of axolotls, and the Aztecs ate them. It was a major source of food for the Aztecs because there were so many there. And it was an ideal habitat for the axolotls, as I say, because it doesn't dry up every year. And so they evolved uniquely there and were eaten in great numbers by the Aztecs. Okay, so the reason why it's a god is because the axolotls thrived in these habitats and provided a great source of food. They also became included in Aztec mythology and art. So there was a god called Xolotl, shown here, and he was about to be, according to their records, he was about to be sacrificed to keep the sun and moon revolving, like typical in old situations. And he decided this was not a good idea. So he transformed himself into a water god called Axolotl, okay, because he was called Xolotl. And so I'm suggesting this is the only model organism in the world that's also a god. And here is an Aztec stone carving of Xolotl once he'd become a water god. So the axolotl is steeped in the mythology of the Aztecs. So what happened? Well, the lake dried up. So this is Google Maps that I did yesterday of Lake Xochimilco. There's a Walmart up here, and there's a Sam's Club here. in case you're ever going past there. And it's now been drained and transformed into a series of canals, and the axolotls have disappeared. Very sad. In fact, there's forwards, go. Hey, yeah. Sorry. Okay, this is Lake Xochimilco today. It's been drained, and the Mexicans use it for Sunday afternoon canal riding in these beautiful canals. There's a little bit of old stuff left on the right, but not very much. And here is the disastrous look at their presence endogenously in Lake Xochimilco. So there used to be thousands of them. And since 2000, the numbers have dropped to almost nothing. So there was an attempt to see how many there were in 2019, and they caught one. So basically, they've been wiped out endogenously in Lake Xochimilco, which is a real shame. Partly the result of draining, partly a result of pollution from Mexico City, and also because of a biological experiment to try and clean it up. They put a load of tilapia in the water, which is good in polluted environments, but the tilapia ate all the axolotl larvae, so that was a disaster. So that contributed to their decline. So essentially, there's none left now in the remains of Lake Xochimilco, and the Mexicans are trying to reestablish them by getting axolotls from America, taking them back and trying to put them back in the lakes. But from the research point of view, it doesn't matter because it's been in the labs for 150 years, as I'll tell you now. So how did we find out about axolotls? Fascinating story. A story of European conquest, as always. When any European country went off to the rest of the world to investigate, conquer, whatever, they always took a group of scientists and naturalists with them so that they could plunder the wildlife, if that's the right word, and send it all back to their capital. And that's how the British Museum is full of the most beautiful artifacts from all over the world, because we stole them from the Greeks and the Turks and who knows what. And that's also how the zoos were full up, and the natural history museums were full up of these wonderful animals and plants that people could see, the local people could see. It was a sort of educational program, but people don't particularly like that now. So they had their 150th anniversary in 2014. So how did they come to be in the labs? So this went back to 1816 when the French decided to invade Mexico for the second time, and they launched the Mexican campaign. So they invaded Mexico initially along with Britain, England, and along with Spain. But when the English and the Spanish realized that their major function of the campaign was to take over Mexico City, they pulled out and left the French to campaign on their own. And this was also the time when Cinco de Mayo came about, because the Mexicans beat the invading French army on May the 5th, 1862, at the Battle of Puebla. So this is a sort of local reminder of what was happening in that campaign launched by the French. So they took, along with the armies, they took their naturalists and their scientists along. And from Mexico, from Lake Xochimilco, the French sent back 34 axolotls back to Paris. And they were sent to the Paris Zoo, the Jardin Zoologique de Acclimatation, basically the Paris Zoo, where they were housed, and 6 of those axolotls, 5 males and 1 female, were given to the Museum d'Histoire Naturelle in Paris, which was nearby. And this guy, Auguste Dumeril, was the one that first started getting axolotls going. Just out of interest, well, actually, a rather frightening interest, at the Paris, this is the Paris Zoo where they were sent back to, and at the same time, all throughout the late 1800s, strange people from Africa were also exhibited, in cages, I would add. And here are some Somalis that people brought back from expeditions abroad. And here are some Hottentots that were brought back. And as soon as they brought back a new group of people and put them in cages, the attendants went through the roof because everyone wanted to see these strange people brought back from different parts of the world. So I thought that was a rather horrendous side issue. And I'm sure they came to look at some axolotls at the same time. So we've got 6 axolotls, 5 males and 1 female from Lake Xochimilco surviving. And these were things of great curiosity. Here's a wood carving from that day showing an axolotl, one of Dumeril's axolotls. And although I've said that you can make them metamorphosed, one of his did metamorphose naturally, and they can seem to have a propensity to be able to do this. And so here's a wood carving of one of those metamorphosed axolotls and a non-metamorphosed axolotls. And these were a source of great scientific debate. Were they larvae? Were they adults? Were they reptiles or were they amphibians? At the time, amphibians wasn't a separate class, so they were classified as reptiles. And then during this debate, they suddenly laid 800 eggs. So that sorted that problem out. They were clearly adults, even though they looked like larvae. And a few of them transformed into land-living animals. So this was the start of axolotls as a laboratory organism. And the first report of limb regeneration, which I and many other people around the world work on, was in 1867, just a couple of years after they'd been given to the Museum of Natural History by Dumeril's colleague called Filippo. It was called On the Regeneration of the Limbs in the Axolotl. Okay, that's a bit too bright. So Dumeril then, after he'd had all these animals laid naturally in his lab, he started giving them all around to zoos, friends, universities, and they spread throughout Europe. So there should be a map here, but here's England's up here somewhere. Somewhere here is France, is Paris. Anyway, they spread, no, Paris is over here. They spread throughout Europe, into Russia, into England, from this one, from these 5, 6 animals that were given to Dumeril. He dished them out all over Europe, and they spread throughout Europe and became famous laboratory organisms because they could regenerate their limbs. And they also started working on the fact on how to make them metamorphose. In 1915, thyroxine was discovered, and axolotls were used as a test for the efficacy of the thyroxine molecule, which people had synthesized in their labs. So they became very important as an assay for thyroxine's potency. Okay, so in the USA, so this is by way of saying, where are they now? In the USA, some of these progeny of Dumeril's original axolotls were given to a famous embryologist at Yale called Ross Harrison. He was sent some from Poland. He gave 5 of these to Rufus Humphrey, who started a colony at the University of Buffalo. Then Humphrey moved to Bloomington in 1957 and started the Ambystoma Stock Center. So that's the Latin name, Ambystoma mexicanum. And he started this stock center, which was funded by the National Science Foundation in 1957. So if you wanted axolotls after 1957, you didn't have to go off to Europe and find some in the lab. You just phoned up Bloomington and said, can I get some axolotls? Then it was transferred to a guy called Antoine Malasinski. And in 2005, when Malasinski retired, the stock center moved to the University of Kentucky and is run by Randall Voss. So if you now want any axolotls for teaching or your research, you phone up the University of Kentucky and order them there. So this is, of course, raises a very interesting question. Don't forget, Dumeril had 5 males and 1 female, or was it 5 females and 1 male? Anyway, that's right. So he had 1 female, 5 males, and all of the world's axolotls have come from that mating. So people are paranoid about inbreeding. And that's really what the current stock center is worried about. But over the years, of course, there have been some reintroductions into this stock center over the period of time. So while it was at Bloomington, there was 10 introductions. When it was at Kentucky, there was 1 introduction. But now there's no axolotls left in the world, so you're completely stumped. Okay, you can't introduce new genetic material to keep your colony from dying out due to inbreeding. And every now and then they have a mating at Kentucky and all the embryos die. So clearly there is a little thing going on. They're worried about inbreeding depression and the accumulation of deleterious mutations over time, which decrease fitness. But there's nothing you can do about it now because they're gone from the wild. And that's probably because they only evolved in one place. But, so this is how we keep them now. I used to keep them when I was doing my PhD in little plastic containers, and I cleaned them out every couple of days. But now you can't get people to do that. But these flow-through systems have been invented for keeping zebrafish in the lab. And so we adapted them for keeping axolotls. So there's a whole series of tanks on racks, and the water comes in one side, drips into the tanks, overflows into the drain port at the back, circulates round, gets cleaned up, and you can keep them in here for months without doing anything at all. You regulate the pH and the temperature and the cleanliness, and it's all very easy. All you have to do is feed them, and they're all tidy and healthy. You have to, so now, so we've been battling. Have you ever heard of IACUC? It's the regulatory authority for animal care. So we battle with IACUC continuously. So if you keep axolotls together in 2s or 3s or 10s, they will bite each other's legs off. Okay, that's what happened. But they grow back, so who cares? So then we went to keeping them on their own because we want to use them for limb regeneration experiments. So we keep them on their own. But then IACUC said, no, we can't keep them on their own because they need to be socially enriched. No. So what is a socially enriched axolotl? So now we have to keep them in pairs. And so to solve that problem, I put tubes in there so they can get away from each other and maybe they won't bite each other. Anyway, those are the sorts of IACUC issues that keep me amused and actually keep me pretty annoyed most of the time as well. Okay, so the first slide was a picture of a white one. With the normal color is green. Here, this is a so-called wild type color. That's what you find them if you ever find one near Lake Xochimilco. Here's a white one. It's called white and not. Here's an albino one. So the albino has no black anywhere, no melanophores anywhere, so it doesn't have black eyes. This guy has black eyes because he's white, not albino, and they have some melanophores, black coloration elsewhere as well. So there are these three genotypes which were very useful for experimentation, and I'll mention that in a moment. So what are they used for? They've been used over this 150 years for studying development. They have a slower development than a frog, and they've been very useful for that. I said they've been used to study thyroxine and how it works, and they've clearly also, their major use is in regeneration because they can regenerate anything. I don't know of anything that doesn't regenerate. Oh, their heart also regenerates as well. I probably didn't mention that. So if they can stay alive during the period of regeneration, they can regenerate everything. And that's why a lot of people use them. And you can see if you amputate the limb here, it gets a little bud on the end and it grows round and replaces all the tissue you've cut off, including all the digits, and everything comes back beautifully. So the other really useful thing about them is they have a much less well-developed immune system than we do, which means you can graft tissues between different animals. So you can graft, you can take whatever you want to do. If you want to do an experiment where you look at the fate of muscle cells, you can graft muscle from a wild-type axolotl and graft it into an albino, look where the cells go, and you will not get a rejection phenomenon. This has been really useful for looking at lineages of cells during the regeneration process. It wasn't very good just using those three examples, white, albino, and wild type. But in the last 10 years or so, 15 years, when molecular biology has come on board, even to these creatures, the genome has been sequenced. It's got a genome like all salamanders do. This guy has a genome 10 times bigger than the human genome. So that was a real problem for molecular biologists for a long, long time. But now it's been done. It hasn't been fully annotated, but it's at least been sequenced. And so all the molecular biology came along associated with this genome sequencing. And now transgenic animals have been made so that you can do beautifully these transfer of tissues and look for lineages. So here's some, here's a red fluorescent protein transgenic axolotl that's all red. Okay, here's the first transgenic that was made, GFP, green fluorescent protein. So every cell fluoresces green in this animal. So now you can transfer your green tissue into your red animal, and you know exactly where the cells go to and what makes what. And you can not only do whole animals, you can do individual tissues which glow green or red by using promoters for specific genes. So here's one where the lining of the brain and spinal cord is green. And here's one where the cranial nerves are green and the brain is red. So you can do any of these kind of tricks that you want once you've sequenced the genome and found the promoters and done whatever you like. So we've made a few of these transgenics in the last few years, but my aim is to make this transgenic a double transgenic, which we're going to call the gatorotl, but we haven't quite made it yet. It's quite easy to get stripes, but we can't get two stripes that don't overlap. That's my current aim. Okay, so here's just a flavor of what we've been doing with these axolotls and the sort of things you can do with them because they're so regenerative in the next 10 minutes. So we studied blood cell development and asked what's the origin of the blood in a developing axolotl. It turned out they had no bone marrow like us, so it wasn't the bone marrow. And to do that, we took embryos. So embryos are amphibian embryos are fantastically flexible. You can cut one in half and graft it to the half of another one. You can make a Y shape if you want. You can graft tissues between them, partly because they're embryos, but also because, as I said before, they don't have a very powerful immune system. So we took the posterior half of a red transgenic animal, grafted it to the anterior half of a green transgenic animal, and made a red-green hybrid like this, and then asked what color are the blood cells? So if the blood cells were red, they would have come from the posterior half of the animal, and if they were green, they would have come from the anterior half of the animal. And in this way, by doing various combinations, we found that the liver was the source of blood hematopoiesis, which is interesting from a developmental point of view. And that was really nicely shown. We did that with these transgenics combined with the ability to be able to transplant tissues. So the other thing that we've had a look at in the last few years is the ability to regenerate a brain, okay, which I think is quite impressive. So here is the axolotl brain dissected out from an animal. This is the olfactory nerve. Here are some cranial nerves. Here's the forebrain. It's a bit like if you take our brain and you pull the cortex off forwards and you stretch the whole brain out. These brains, and fish brains as well, are in a row. Our brain is like piled on top of each other and with the cortex. If you stretch it out, you get the lizard, amphibian, and fish brain. So here's the forebrain, here's the midbrain, here's the hindbrain. And it was shown in the 50s that if you can take out the whole forebrain, and it will regenerate in about 2 months, which is pretty impressive. And it will regenerate partly from the olfactory nerve, which you've left in place, and partly from the cells remaining at the back. So we've done a series of experiments to try and understand what cells are responding during brain regeneration with much less of a damage. So we just cut the top of the forebrain out. We just cut this oblong out here. You can see this is the blood vessels in the middle of the forebrain here, the choroid plexus. We take quite a lot of the brain out, and over a period of time, in 34 days, you've got just a little bit of a damage here, and in 40 days, you can't see any damage at all. And if you section through that, you can see exactly that the brain has been completely replaced, including all the tracks that come across the forebrain. The whole thing is regenerated, which would be fantastic if we could extrapolate that. And we're trying to find out the cells that give rise to the cells that replace them. Using this transgenic animal here. So this transgenic animal labels the lining of the brain, the ventricle lining called ependymal cells, green. So if we do experiments on this guy, we can see if all the tissue which is regenerated is green, in which case it came from the ependymal lining. If it's not all green, it would have come from neurons as well. So we're trying to do that currently at the moment. Okay, and of course, I've done a lot of limb regeneration experiments over the years. And here's just a little montage of what happens and showing you how quick it is. So here's a white axolotl. You amputate the limb. 3 days, wound healing is complete. 1 week, you get this little bud of tissue called a blastema, which grows on the end. It elongates and flattens. This is called the palate stage. And then you start to see the appearance of digits coming back. And in 3 weeks, you've got it's only got 4 digits anyway. You get a 4-digit nice regenerate coming back in 3 weeks, which is pretty impressive. I've been interested since 1980 when I published a paper on it, on how the cells know where they are in a limb. It's called positional information. If you cut the limb off here of an axolotl, it grows back perfectly. If you cut the limb off here, it grows back perfectly. So the question is, how the hell does it know what's missing? So the cells at the cut in the blastema must have some measure of position. They must know, oh, a whole limb is missing, or just a finger, or just a forearm. And I've been working on that for 42 years because early on I discovered a way of changing the positional information of the cells at the cut stump using a derivative of vitamin A, or the active metabolite of vitamin A, called retinoic acid. That's widely used in the body. With all the vitamin A organs that we have use retinoic acid. It's the potent molecule. It's the derivative of retinol, which Olay puts in all their face creams. So I found a long, long time ago, if you amputate the limb through the carpals, through the hand, it will regenerate exactly what you cut off. If you treat with retinoic acid, you can fool these cells into thinking they're actually back here at the shoulder. And instead of regenerating just your hand, it'll now regenerate a whole limb from the hand level. And we've just published a paper where we did RNA sequencing and identified the genes that are up and down regulated during this process of respecification of the blastemal cells. And there were 117 up, and I mean, there were 1,170 up and 1,340 down. So we still got a long way to go in my long-term experiment. But we're making progress on understanding exactly what is the mechanism of how cells know that's where they are in these tissues. And the other thing that we're looking at is trying to understand the cellular origin of the differentiated products of the blastema. So does the muscle only make the muscle in the new limb? Does the bone only make the bone in the new limb? Does the epidermis only make the epidermis? What's the sort of relationship? And a long time ago, it was shown that if you dissect out all the cartilage or the bone in a limb, in an axolotl limb, and stitch it all up, and it like flops down below, it's got no bone in it, and then you cut it off, the thing that regenerates will be complete. It will have new bone in it and new muscle and everything. So it's quite an interesting concept, what the lineage specifications of these cells. And we've been doing it using these GFP transgenic animals by grafting between animals. So, for example, if you take a GFP limb and graft some skin, dermis and epidermis, in a cuff on a white animal, then cut it off and ask, okay, what do these green fibroblasts from the dermis make? And they spread out through a hole, the regenerate. Here's where it was amputated through the graft. And here is where you can see the green cells from that cuff of skin and ask what they've made. It turns out they make cartilage, more dermis, fibroblasts, tendons, all of these fibroblastic type of cells, but they don't make any muscle at all. So there's quite flexibility in the fibroblastic lineage in the blastema. Similarly, if you graft a piece of cartilage from the GFP animal into a white animal, amputate through the graft, let the animal regenerate, see what happens. There's plenty of green cells in the regenerated tissue. And it turns out cartilage can make more cartilage, can also make tendons and dermis and fibroblasts and ligaments. So there's this flexibility in this group of cells that during regeneration. And lastly, if you take muscle, and you make these quite complicated experimental constructions, but you end up with a limb that's white but has green muscle in it. Then you amputate the limb. Now you ask what happens, and it turns out muscle only makes muscle. Okay, this is a very strict, interesting lineage. So if you take the muscle out of a stump of a limb, amputate it, the regenerate will not contain any muscle. And interestingly, muscle is the only cell that's got a lot, the only tissue in the limb that's got a lot of stem cells in it. So they seem to be very lineage specified in an interesting way. So we're quite interested in how these cells can, or at least the fibroblastic cells, can change their fate, because if we can do that at will, we can make some interesting changes to the tissues and how they're differentiated. Okay, so we're interested in, this is just the last cell slide now. Can cells change their fate during limb regeneration? It would be really interesting to be able to do that. And what's the contribution of stem cells to limb regeneration? And of course, everybody wants to be able to induce limb regeneration in a mammal. It turns out there's a little bit of potential already there. And this was discovered in the 30s. So here is a section through a mouse digit. This is the last phalange here, and the mouse has a claw on it. And here is the phalange before the last phalange. It turns out that if you cut halfway through this claw and last phalange, it will regenerate very, very nicely. And this is a regenerate, this is the original. Nice regeneration if you cut halfway through the mouse digit. And it was known from these axolotl studies that if you cut the limb off and then stitch the skin up very tightly over the wound and stop epithelial coming in, epithelial wound healing, you inhibit regeneration. That's been known since the turn of the 1900s. And finally, a surgeon in England in 1976 said, oh, well, I'm going to try this in my children's cases. So before 1976, or maybe still now, depends on the surgeon, if you're a child and you have your fingertip accidentally amputated, before then, the surgeon would stitch the wound up completely tightly and make it look therapeutically nice. If your missing tip looks therapeutically nice, but it doesn't look a mess. And we already knew that if you did that to an axolotl, it'll stop limb regeneration. So this person called Illingworth decided to leave the fingertips and just put a cocoon on the top, and they all regenerated. So here's a rather bright picture of one. So here's a child. This turns out to be a 5-year-old girl with the digit tip amputated in an accident. And here it is. It looks fantastic. Perfect regeneration of the nail and half of the finger if you just leave it and put it in a cocoon and let it do its own thing. And that's why many people study mouse digit tip regeneration, because it mimics humans. Apparently it works up until the age of 12. After that, we tend to lose the ability, although there's the occasional report there was one in CNN where this lady did regenerate her fingertip. She was like 45 or something. So maybe it's sporadic. But this is what we would all like to do. Find something either from mouse or axolotls to make this a prolonged state into adulthood. Of course, if you cut further back, you don't get any regeneration. It's only this end phalange that works. So we all want to try and induce this in mammals by understanding this. And there's a big program at Texas A&M. If you cut off here, as I said, it won't regenerate, to try and induce regeneration of more than just this digit tip. And so far, this guy, Ken Monioka, has got all the way down to the end. He's made a new joint, and now he's trying to start to do the next phalange as well. So it's quite humanly relevant, the sort of things that we've been doing. And I'm in good time, and I think that's my last slide. Yes. Okay, so we're trying to extrapolate this to humans, basically.
So are there questions?
The lifespan of these animals.
Oh, they're round about 10 to 12 years. Yeah, they last a long time, and they continue, like fish, like many animals, lower animals, they continuously grow. So I've had them this big, and I've had them 10 years old. But other salamanders, so there's a giant Japanese salamander that's 30 years old. It's in the Tokyo Zoo, and it regenerates. They tried it. They regenerated its limb, but it took about 5 years to regenerate. But it did it just slowly. So the lifespan can be enormous, partly because they continuously grow.
So the fact that they continuously grow implies that regeneration doesn't slow with age, or does it?
Slows
It
with
does
age.
does slow with age,
Yes,
even
it
though they're
does.
still growing.
Yes, it does.
Okay, and then
A lot of people think as you get older it stops, but it doesn't. So the Japanese giant salamander still regenerated its limb, even though it took 5 years to do it.
Okay, 2 more questions.
Of course.
One, you were talking about regeneration with amputation, but you also removed the bone. It won't regenerate that bone, even though the limb is still there?
No.
So it really requires that blastema.
Exactly.
Yeah.
Yes,
And
spot on.
Then the other question is it sad about the axolotl? I was wondering if there's any relationship to the lungless cave salamanders that sort of look a little bit like it.
Oh, yes. Well, they're all fairly closely related, and they've all come from these very, I don't mean obscure, from these fairly obscure places, and they're evolving almost in isolation. Yeah, exactly. I don't know that, no, but I know of no salamander that does not regenerate. I haven't seen a report on it, but my guess is it must do. So people have been scouring all the salamanders in Florida and North America and wherever, and they all regenerate.
I just have 2 points. One is I love your shirt. I want to know if one of your salamanders modeled for that. And the second is, since especially here at OCAMIC, we'll be very interested in your brain study because we'd all like that, and so we'll have to follow that. Thank you.
Well, I've got to try it on myself first, haven't I?
Are there any questions in the chat or among the Zoom people? No, I guess not. I don't see anyone. Any other questions here? Do you have one, Beth? No? No? Okay. Yeah, okay, Lynn.
Thanks. I love that talk. I have to say it was excellent. What are you working on now? Are you still working on axolotls, or have you switched to another animal?
We're still, about to buy some more axolotls and get it going again. It was only last year that I stopped working on axolotls with the publication of that retinoic acid paper. But I've also been working for the last 10 years on spiny mice, which, as Jane mentioned, is a mammal that seemed to be able to regenerate skin, spinal cord, lots of things, muscle, maybe heart. So I'm actually now at this very moment working on heart regeneration in spiny mice after a myocardial infarction. So they seem to be able to do that. But we're going to just get some axolotls again and use them for a sort of Alzheimer's type research to see what happens to the axolotl brain when you inject phosphorylated tau into it. That's the nasty thing in Alzheimer's. So if you inject it into a mouse brain, you give the mouse Alzheimer's. We're going to see what happens if you inject it into a axolotl brain.
Oh, interesting.
So
we heard a talk this morning that dealt with injecting tau into a mouse.
oh, really? Oh, that must be from, that was from you. Oh, yeah, nice.
Yeah, I was
Yes,
telling
you were
you
saying
about
about that.
How expensive is an axolotl? You said you had to buy some more. How expensive is it?
They are $5 for about this size. They're not very expensive. You can buy eggs for 25 cents each and grow them up yourself, which I do every year for my students. I'd say that was fairly cheap. But there's 2 available in Aquatropics on 34th Street, and they're $99 each. So don't go there. Come to me, I'll give you some.
Did you say they're reintroducing them into the wild in Mexico?
Yes, they are. There's a big program to try and do that. But they've got to battle all the Mexicans on Sunday partying. Pollution from Mexico City still continues. So I don't know whether they'll be very successful. Maybe they'll go to a different place, which might be less polluted, I don't know. But because they're not around now, they've been designated a highly endangered species by that CITES organization. So that, and that's a complete nightmare now to send some axolotl. So the stock center, for example, will send them all over the world, like to Europe and wherever you want to go. But you can't do it now because it's an endangered species. And they're tearing their hair out. Yeah, but look, I've got thousands of them here. But because it's the same it's the same species name, they've stopped being allowed to be to send. So they're trying to change the name of the species at the stock center so that they can be freed from the goodwill of CITES organization, which has had a knock-on effect. So,
Well, they could also use legislation to exempt those animals. I mean, that'd be another way to go about it, I suppose, but
yeah,
complicated.
yeah.
Yeah. And yeah, would require some cooperation. Well, are there any other questions? Anybody have anything you want to say? All right. Well,
thanks
thank
for
you so
coming.
much. This was great.
Thanks for coming.